Biotechnology is basically humans saying, what if we edited life? It involves using biological systems to make useful products. This includes medicine, forensics, and agriculture. Most modern biotechnology focuses on DNA. Scientists analyze, copy, and modify genetic material. PCR, gels, and CRISPR are the greatest hits. And yes, exams love them deeply. Quick heads up, if you stick with me until the end, I'll go over the biotechnology questions that show up consistently on AP and IB exams, plus test prep tips that help you avoid the most common mistakes. Yes, this includes PCR, gels, and CRISPR, and yes, your exam absolutely cares. Biotechnology questions appear everywhere on AP and IB exams. Memorization alone will not save you. You must understand how each technique works. Exams love diagrams and experimental setups. Data interpretation is extremely common. Ethical questions sometimes sneak in as well. Understanding biotech boosts multiple units at once. PCR stands for polymerase chain reaction. It is essentially a DNA photocopier. Starting with tiny amounts of DNA, PCR makes millions of copies. The process requires primers, free nucleotides, and a DNA polymerase. A thermal cycler controls temperature changes. PCR mimics DNA replication, but much faster. This technique shows up constantly on exams. PCR has three repeating steps. Denaturation uses heat to separate DNA strands. Annealing allows primers to attach. Extension builds new DNA using polymerase. These steps repeat over and over. Each cycle doubles the DNA amount. After many cycles, you have a lot of DNA. Gel electrophoresis is basically a DNA obstacle course. DNA fragments are loaded into wells in a gel. An electric current is applied. DNA moves towards the positive electrode because it is negatively charged. Smaller fragments travel faster. Large fragments lag behind dramatically. The final pattern creates visible bands. Reading gels is a favorite exam activity. Each band represents DNA fragments of the same length. Smaller fragments move farther down the gel. Larger fragments stay closer to the wells. A DNA ladder provides size reference. Matching band patterns can identify samples. Exams expect confident gel interpretation. Restriction enzymes are the scissors of biotechnology. They cut DNA at specific sequences. Each enzyme has a recognition site. Some cuts create sticky ends. Others produce blunt ends. These enzymes are essential for DNA analysis. Bacteria invented them first, because bacteria are clever. CRISPR is the most dramatic tool in biotechnology. It allows scientists to edit genes directly. A guide DNA targets a specific DNA sequence. Cas9 cuts the DNA at that location. The cell repairs the break, creating changes. The system involved in bacteria as defense. Exams love CRISPR questions. CRISPR has powerful applications. It is widely used in research. Medical treatments may target genetic diseases. Agricultural organisms can be modified. Ethical concerns are important. Germline editing is especially controversial. Exams often ask students to evaluate benefits and risks. Biotechnology tools often work together. DNA is extracted first. PCR amplifies the target region. Restriction enzymes cut the DNA. Gel electrophoresis separates fragments. Scientists analyze the results. CRISPR may then modify genes directly. Biotechnology questions are very predictable. PCR steps are frequently tested. Gel interpretation appears constantly. CRISPR mechanism questions are popular. Data analysis is common. Exams test application, not memorization. Clear explanations earn points. Always use precise biological language. Examine steps in order. Refer clearly to diagrams. Avoid vague answers. Connect tools to their applications. Address ethics when asked. Practice with real data and diagrams. If you follow this advice, you are going to ace all of your tests. Good luck. That's it for now. Don't forget to subscribe, like, and check out Dr. B's tutorials on Amazon for full AP and IB study guides. See the first link in the comment below. For more tutorials and test prep videos, subscribe to our YouTube channel, Dr. B's Tutorials.